Hello everyone. So today's lecture is basically focused on the few important factors of micellar structures. So let's first try to discuss about few distinct features of the micelles. So firstly, uh, we can identify that there is a presence of a strong intermolecular interaction between the hydrocarbon chains of the micelle molecules, that means the surfactant. Okay, so obviously there are long chain hydrocarbon molecules present, and the intermolecular interaction between these long chain hydrocarbon is so strong that those hydrocarbon molecules almost behave like liquid molecules. So that means the interactions between two liquid molecules are almost they almost become identical with the interaction between two hydrocarbon chains. So that means the interaction is so much favorable between the hydrocarbon phase. And the second distinct feature is, once again, we see strong intermolecular hydrogen bonding between the polar head groups and the polar medium, mostly like water. Okay, because mostly the micelle formation is studied in water as the dispersion medium. So strong intermolecular hydrogen bonding between polar head groups and water, and this hydrogen bonding is so pronounced that sometimes it also promotes strong water molecule interactions as well. All right. So this is mostly common for any types of micelles that are formed in water. Now the question is, which experimental tool helps us to find such distinct characteristic structural features of micelles? So we can write that experimental tools to study micelle structure is, one of them is definitely dynamic light scattering, DLS, that I have already talked about it while I discussed about the microscopic uh, electrophoresis, that is particle electrophoresis, often coupled with the dynamic light scattering, that is the laser Doppler effect. And also there is something called low angle X-ray diffraction. Okay, so sometimes this, by some special experimental technique, the micellar phase can be isolated. Okay, so that means it can be isolated in a frozen solid form, and then doing some low angle X-ray diffraction. So this low angle means the scattering angle lies between, say, fractional means fraction of 1 degree, and then maximum up to 2 degree, 3 degree. I am talking about 2 theta values in powder X diffraction. So in this low angle, certain peaks are recorded, and from those peaks we can try to understand how the orientations or the structures of the micelles are. So now we'll move on to a topic on critical micelle concentration, CMC. So the topic of the next discussion will be, what are the important factors that actually govern or affect the critical micelle concentration. So let me clean the board and start the discussion from here. All right, so let's do that. So primarily there are 4 governing factors that affect the CMC. So first of all, we have say up to carbon number 16. That means up to 16 carbon chain. So we can think about cetyl group, okay, C16. The critical micelle concentration is linear with the carbon chain length. So that means, suppose if I have a C10 chain, that means the surfactant molecules whose hydrocarbon chain consists of 10 carbon atom versus C12 molecule versus C14 and versus C16. If we make a series of these four surfactant molecules, then we will see the CMC would gradually change from C10 to 12 to 14 to 16, all right, by following a linear fashion. So the question to you is so in which end you think the CMC is lower or higher? Say if we compare between C16 versus C12, so where would you think that CMC would be lower or higher? Means for C16, would the CMC be lower or higher compared to C12? All right, so you just think about it using your own understanding, and then try to arrive at a conclusion. All right. So next thing is the thermal agitation. So would thermal agitation is good for making of micelle? Obviously no, because if you keep on disturbing the medium with strong heating as well as the mechanical stirring, so it hinders the formation of micelle. Okay, so that means it would be very difficult to find a CMC, to achieve a CMC, if you have a thermal agitation existing in the medium where you are going to make the micelle. And then we have some stabilizing agents. For example, if we are talking about some ionic surfactants forming micelle, so in that case, sometimes what happens if we add some electrolyte, so they actually electrolytes, they help to form the micelles by screening the charge of the polar head group. So the polar head group might suffer from strong repulsive interaction between the other head groups like this. So we can think about, say for example, we have a spherical micelle. So I am just drawing a part of the spherical micelle like this. Okay. So this is the spherical micelle. This is the part of the sphere. And assume that the polar head groups are negative. Okay. So these negative charges may feel a strong electrostatic repulsion between the light charges. So in the meanwhile, if I add some positively charged electrolytes, there means some addition of electrolytic ions. So these positively charged ions may screen or may seep into the structure, and they can fit in between, and they can screen those repulsive negative charges, and they act as a structure stabilizing agent. So it imparts a favorable interaction. All right. So for ionic surfactants, charged electrolytes sometimes act as stabilizing agents. And for non-ionic surfactants, sometimes also, sometimes it also acts for ionic surfactant, that is sugar molecules, because sugar molecules are famous for making strong intermolecular hydrogen bonding. So sometimes they help to make a favorable interaction between the polar head groups and the water. So sometimes adding sugar molecules helps to form the micelles. So you will have a lower CMC if you have these things present as stabilizing agents. So that means if these stabilizing agents are there with a relatively lower concentration, you can achieve the critical micelle concentration. So, and another thing is, the last thing is the destabilizing agent. So these destabilizing agent are also known as structure breaker. Okay, so and these things can known as structure maker. So the structure breaker molecules are typically known as urea, and also it could be formaldehyde. So if you add these things, and if you can recognize these molecules, these are also known as effective denaturing agents for protein. So if these molecules are there, they can help to break the micelle structure, and definitely with a small amount of this destabilizing agent, you will need a very high amount of surfactant to get the micelles. So that means your CMC will be higher. All right. So these are some general information about how to control or tune the CMC. And then we have to learn about one definition which is very relevant in the topic of micelles or critical micelle concentration. It is called the Kraft temperature. So let me clean the board and let me discuss what is Kraft temperature. So Kraft temperature is an important parameter for micelle formation. So it is a temperature above which the hydrocarbon chain behaves as liquid. So that means above the Kraft temperature, it is possible to form micelles, and below it, it is not possible. So you can take an example of say whenever we have long tail hydrocarbon chain. So think about some oils or fat, like think about coconut oil. Normally in winter, the ambient temperature is cool enough to make the oil solidify. All right, and once the summer comes, the ambient temperature goes up, and we have the liquid form of the oil. So it's similar, something like that. So for example, for ionic surfactant CTAB, the Kraft temperature is approximately around 20 degrees Celsius. So that means above 20 degrees Celsius, it is at all possible for CTAB to form micelles in water, and below 20 degrees Celsius, it would almost behave like frozen substance, and it won't form the micelle. Okay, so you need to have some liquidation effect of the hydrocarbon chain to form micelles. So therefore, the temperature that defines the transition is the Kraft temperature. So above the Kraft temperature, the hydrocarbon chain basically behaves as liquid and capable of formation of micelles. But one thing we should also remember that for certain ionic surfactants like CTAB, if you increase the temperature above Kraft temperature and slowly raise the temperature, it does not hinder the micelle formation process. So maybe the higher the better, the higher the temperature, the better the micelle formation. But for non-ionic surfactants, as I said earlier, the high temperature, like thermal agitation, higher temperature is not good. So at higher temperatures, many non-ionic surfactants, the polar head groups becomes less hydrophilic, and they can precipitate out from the medium. So anyway, these are a few small details about the formation of micelles and critical micelle concentration. So now we are going to study the different possible types of micellar structure. All right. So let's erase this part, and let's start the topic right from here. Okay. So on the board you can see the 5 different types of major micellar assembly, and some useful information on the right hand side. So let me tell you first that there are 5 major types of micellar structure: spherical, vesicular, lamellar, hexagonal, and a bicontinuous cubic phase. can be obtained. And all these structures can be predicted by a formula, and this formula is known as critical packing parameter, CPP. And remember, this is a purely empirical formula with some mathematical calculation is taken care into. So the CPP expression is equal to, all these structural features can be predicted that which micelle is formed on which characteristic features. So is equal to CPP is equal to V divided by A naught multiplied by LC. So V stands for volume of the hydrocarbon chain, A naught stands for the head group area of the polar head group, and LC stands for the critical chain length. And here how V and LC is expressed in terms of N and M is given. So N is the number of carbon atoms in a particular hydrocarbon chain, and M is the number of hydrocarbon chain present in the surfactant molecule. Remember, the surfactant molecule may have more than one chain because it could be branched chain. So there could be 2 or 3 chains. So usually for a molecule like CTAB, M is equal to 1 because it has got only one hydrocarbon chain, and N is the number of carbon atoms in the hydrocarbon chain. Okay, so this is how V and LC depends on M and N, and so this volume unit is in the angstrom cube, and this critical chain length given is in the unit of angstrom. Alright, so if we plug in all those values, we get a particular number for CPP, and usually CPP comes into fraction. So the fraction can come anywhere between 0 to 1/3, or from 1/3 to 1/2, and then 1 and beyond 1. And so whenever the CPP changes from more than 0 but less than 1/3, so in this region mainly spherical micelle has been seen to be formed. And then between 1/3 to half, some polydispersed cylindrical micelles, cylindrical micelle are formed. And you may wonder that why these cylindrical micelles are not being mentioned here. I will talk about that because it is already included in one of the structures. And beyond half, between half and 1, we can see some vesicular structure, okay, and also some bicontinuous phase is there, and beyond 1 we get inverted structures. Okay, means the structure just inverted because once the value go beyond 1, so that means the V volume of the hydrocarbon chain is very high, very big compared to the polar head group area, and multiply the chain length. Alright, so now we get to see how these 5 different structures are described. Okay, so let's discuss it one by one, starting from the structure of the spherical micelle. So let's do it. So let's first with the spherical structure, and before drawing the structures, let's assume that the dispersion medium is water. So therefore, we will be considering only the normal micelles. Okay, so that means the hydrophilic group would be facing towards the water medium, and the hydrophobic group will be buried inside the micelle, the core, means in the pocket. So the spherical micelle, this can be regarded as a cross section of the micelle. So it is all around like a flower, spherical. And so one very important feature is the inside here, the core, there is no space. That means the inside is not empty. It is absolutely packed up. Okay. And secondly, mainly anionic surfactant. Means all the detergents, usual detergent soaps that you find, you will invariably find the anionic surfactants. and they like to form the spherical micelles above the critical micelle concentration. And next we go to the vesicular micelles. So just one thing, just notice the spelling VESICULAR, vesicular. By mistake I wrote a C, earlier. So please make a note and please correct it. So the vesicular micelle is actually it will form a vesicle, means a hollow structure. Okay, so it will go like this. So we can think that it is a kind of a bilayer structure. So inside this is hollow, and outside, assuming that the medium is water, then normal micelles. So we have the polar head groups like this, and the non-polar chains would be facing each other. So this structure would continue on both sides, and therefore it would form a hollow vesicular micelle. Okay, so this vesicle actually also very much like the cell membrane, right? The lipid membrane. So the lipid molecules also arrange in a similar fashion. So if we enlarge this portion, so it would look like something this. So this is the hydrophilic ends, and in between we have this long tail hydrocarbon chain. So this is the hydrophobic core. Okay, so we can actually think it like this. So this resembles the cell membrane structure. Anyway, so next we go to the lamellar structure. So this is vesicular structure, this is spherical structure, and we are discussing the normal micelles. So in the lamellar structure, the shape is almost the sheet-like structure. So you can extend the vesicular structure in a two-dimensional sheet, then it will become a lamellar structure. So something like that. By schematic diagram, we always represent micelle something like this. By circular structure means polar head group, and long chain is represented by some straight lines. So now if we think that as a 3D structure, a 2D structure, so we have the micelles, the surfactant molecules lying around here. So the structure is a two-dimensional flat one, and this would continue. So here you go. So it looks like a two-dimensional sheet. Okay. So this is a lamellar structure. And then we have this hexagonal structure. So in hexagonal structure, the surfactant micelles undergo hexagonal close packing. So this is also known as hexagonal close packed HCP structure of the micelle. So how it goes is something like that. So actually, the surfactants form a cylindrical micelles. Okay, so here now we are talking about a cylindrical micelle. So it is not exactly spherical. So cylindrical means, so this would actually, this is a three-dimensional structure. It goes like this. So the micelles, the surfactant molecules are packed in a form of a cylinder. Like outside you have this polar head group, all right, like this, and inside we have the hydrophobic core. And if we take the cross section of it, so this is the circular cross section we can see. So the circular cross section resembles the cross section of the spherical micelle. But where is the hexagonal close packing? Now the hexagonal close packing appears that when 6 such cylinders pack around one cylinder. So 1, 2, 3, and we have 1, 2, 3, 4, 5, and 6. Okay, so now if you think about that, so this is a bundle of such cylinders held together, and remember, in each of these cylinders represent the cylindrical or hexagonally packed cylindrical micelles like that. So an assembly of 6 such like 7 I would say. So one, so okay, so this is the hexagon, and in the center we have one cylindrical micelle. So an assembly of 7 such cylindrical micelles form the hexagonal closed packed micelle. So this is a very distinct structure of the micelles, and many surfactants, including CTAP, form this kind of assembly. Okay, so this is a very interesting structure. And the last one is the bicontinuous cubic phase. All right, so the bicontinuous phases appear when, suppose, a micelle. So a bilayer is formed like this. It's almost like the wall of the vesicular or lamellar structure. So the building unit is very much like lamellar one or the vesicular one. So like this. So now this is just a part of the the bigger micelle structure. But think about this. If this micelle structure actually goes in a zigzag fashion like this, all right, and continuously, so there is no end to it, and this zigzag structure almost follow the DNA double helix like twists and turns, and in 3 dimension they form a very distinct structure. So it takes a shape in such a manner that in every twist and turns they form some hollow spaces. So that hollow spaces very unusually in 3 dimension pack up in a particular manner, and in this case it usually packs up with a cubic symmetry. So that's why it's called a bicontinuous cubic phase. So indeed from my description you can understand that this is indeed a very complicated structure, and it is very difficult to draw in a 2 dimension. We can only generate these drawings through computer 3D diagrams, but the bottom line is a bicontinuous structure, how to describe. So this bicontinuous structure, it takes up the 3 dimensional space with some twists and turns in such a manner so that the void spaces inside holds a 3 dimensional cubic geometry. All right, so that's all about the 5 distinct types of micellar structure or the surfactant self-assembly. All right, so with this we finish our lecture topic on the surfactant and micelle. So I will see you in the next lecture, lecture number 14, which is on emulsion and microemulsions. So till then, goodbye.